When talking about the anaplerotic reactions, those reactions are a specific set of processes that are responsible for balancing out the concentration of the intermediates found within the TCA cycle. So remember that the TCA cycle has intermediates, and the entire process is a catalytic cycle. Since it's a catalytic cycle, all intermediates must be maintained at specific concentrations in order for the TCA cycle to work properly. If the concentrations of the TCA cycle are not kept constant, the TCA cycle will not be able to occur properly. So, the anaplerotic reactions are responsible for balancing out the concentrations of the intermediates in the TCA. So, one of the anaplerotic reactions involves the enzyme known as pyruvate carboxylase enzyme. Now, this enzyme catalyzes the reaction between pyruvate and oxaloacetate. When we go from pyruvate to oxaloacetate, we utilize biotin as well as a cofactor. We have ATP as a cofactor, and we're going to see the release of ADP and inorganic phosphate. Then we also have H2O and CO2, which are going to combine to form bicarbonate. All right, now let's look at the mechanism. So, in the first step of the pyruvate carboxylase mechanism, we see that first of all, we already have the conversion of bicarbonate. So bicarbonate is just the interaction between water and carbon dioxide to form the following molecule right over here. Now, biotin is a cofactor that is going to be bound to pyruvate carboxylase. So pyruvate carboxylase is an enzyme, and enzymes is made up of proteins, and proteins are made up of amino acids. So essentially, pyruvate carboxylase will have amino acids. Specifically, pyruvate carboxylase has the amino acid lysine, and lysine is going to bind with the biotin. So we see this amide bond right over here between the lysine and the biotin. The biotin is this structure in blue over here. So the biotin, once again, forms an amide bond with the lysine, which is found within pyruvate carboxylase because it's an enzyme. Enzymes are going to be made up of amino acids. So when biotin is bound to pyruvate carboxylase, it basically acts as this transporter molecule, and we'll see how the transportation works. But the first thing we have to make note of is that this first reaction that is going to happen is going to be happening in the first catalytic site. Pyruvate carboxylase has two catalytic sites. This first reaction we're going to talk about happens in the first catalytic site. So we have bicarbonate right over here. And bicarbonate is going to have this oxygen with an extra lone pair, so it has a negative charge. That bicarbonate is going to act as a good nucleophile, and it's going to attack the phosphate right over here. So it's attacking the terminal phosphate on ATP. So the structure in orange over here, this is the structure of ATP. Now when it attacks that phosphate is going to have to lose one of its bonds. So this bond over here is going to break. And the oxygen over here is going to gain a lone pair. And as a result, we end up with this molecule over here. So it's ADP. So it's adenine diphosphate. All right. So we're going to see a release of ADP. So bicarbonate is then able to form a bond with that terminal phosphate on the ATP. And as a result, we get this intermediate known as carboxyphosphate. This carboxyphosphate intermediate is a very short-lived intermediate because these electrons over here in step 2 are going to fall back, and this oxygen is going to form a double bond with carbon. And then since carbon can only have 4 bonds, it must break a bond. So it's going to break the bond right over here, and this oxygen will gain the lone pair. And as a result, we see the loss of inorganic phosphate, because when this oxygen gains that lone pair, we get a structure like this. We see PO, O minus, O minus, O minus, right? So that's inorganic phosphate, and it'll be lost because this carboxyphosphate intermediate is very short-lived. Now, as a result, when the following electrons, when they fall back to form that double bond with a carbon, we get carbon dioxide, CO2. Now, CO2 is in a good position to be attacked by a nucleophile, right? So biotin, this is where biotin is going to come in. And just to reiterate, this entire reaction right now is happening in the first catalytic site. So the lone pair on the nitrogen on biotin is going to nucleophilically attack the carbon on carbon dioxide. So carbon can only have 4 bonds. So if it wants to make a bond with the nitrogen on biotin, it has to break one of its bonds. So it's going to lose one of its bonds with the oxygen. So oxygen is going to gain a lone pair. So down here, we can see right over here that the oxygen gains a lone pair and carbon now has a bond with the nitrogen on biotin. One of the things that we want to make note of is that CO2 is the more active form compared to biotin. So essentially, we want to go from a stable form to a more active form to take part in the reaction. So going back down here. So now that we've had this carbon, the CO2, it's now attached to the biotin. We get this intermediate known as the carboxybiotinyl enzyme intermediate. So remember, this biotin is bound to the lysine on pyruvate carboxylase. So it's bound to pyruvate carboxylase. Now, the next step, so once this carboxybiotin intermediate is formed, biotin will transport the CO2 to the second active site. So up until this point, the reaction was happening in the first active site. But once we get the formation of this CO2 attached with the biotin is going to move the CO2 to the other second active site. All right, so over here, biotin transports the CO2 from one active site to another, and then we can see the reaction happening right over here. So we had the CO2 on this side, now it's on this side. So essentially it moved from active site 1 to active site 2. Now we're going to see in active site 2, we'll see the second reaction that happens. But first we need to make note over here that when oxygen, this oxygen over here, it has a negative charge and it wants to form a bond with carbon, right? So once again, this is going to be a short-lived interaction where the lone pair on the oxygen is going to fall back over here and it's going to form a double bond with carbon. And remember, carbon can only have 4 bonds, so now it has to lose a bond. So it's going to break the bond with nitrogen. So now nitrogen has an extra lone pair. So it's going to form a double bond with the adjacent carbon. But this carbon over here can only have 4 bonds, so it has to break a bond. So that carbon is going to break one of its bonds with oxygen, so oxygen is going to gain a lone pair. So we see that down here. So biotin is decarboxylated, and we see the double bond gained over here, and we see the oxygen has gained a lone pair. And as a result, once again, we see the loss of CO2, right? Because over here, we lost that CO2. Now we're going to go back down. We're going to take a look at what happens next. So now we're still in the second active site. Now we see the interaction between pyruvate and biotin. So pyruvate has on this carbon right over here, it has 3 hydrogens. So remember, biotin over here, it has this lone pair and has this oxygen with a negative charge, and we want to stabilize biotin. So what we're going to do is that we are going to drop down the electrons over here, and then we are going, as we drop down the electrons and oxygen forms that double bond with carbon, we are going to see that carbon over here is going to break one of its bonds. So as it breaks one of its bonds, it's going to break the bond with nitrogen, and then nitrogen will gain the lone pair, and then nitrogen will attack the hydrogen on pyruvate. When it attacks the hydrogen on pyruvate, carbon needs to have 4 bonds, so it's going to form a double bond with the carbon on pyruvate, the second carbon on pyruvate. So when this second, over here, I'll zoom in. So when the second bond is formed, carbon can only have 4 bonds, so it has to break a bond. It's going to break one of its bonds here, and oxygen will gain a lone pair. Okay, this was a lot going on, so I'm going to reiterate some stuff. So over here, we can see negative charge. We want to stabilize that, so we're going to form another bond with this carbon over here. Carbon can only have 4 bonds, so it's going to break one of its bonds, and it's going to break the bond with the nitrogen. So nitrogen now has a lone pair, so it has a lone pair. It's going to nucleophilically attack this hydrogen over here. Nucleophilically, when it abstracts this hydrogen, this carbon needs to still have 4 bonds, so it's going to form a bond with the adjacent carbon over here. Now this carbon can only have 4 bonds, so it's going to break one of its bonds over here, so this oxygen is going to gain a lone pair. All right, so this process results in the enolate form of pyruvate, and we see that right over here. Now this enolate form is very important. That's what we were trying to make this entire time. But if we take a step back and we look at biotin, once again, we've regenerated biotin to exactly how it was in the beginning. Because remember, it's a part of the enzyme. We want to regenerate anything that we're using to catalyze this reaction. So down here, we can see that this is our enolate form of pyruvate. And now the enolate form of pyruvate is going to interact with the carbon dioxide that we formed earlier. So the electrons over here that form that double bond with the 2 carbons is going to attack the carbon on carbon dioxide. So in order for these 2 carbons to form this bond with one another, this carbon has to lose one of its bonds because carbon can only have 4 bonds. So it's going to break one of its bonds over here, and oxygen is going to gain a lone pair. So I'll draw an arrow like this. And then when this, the electrons over here, when they attack the following carbon, it's all happening because we need to stabilize this oxygen, right? Because this oxygen has a negative charge, has a lone pair, so the lone pairs are going to fall back, and we're going to see a double bond form between these, the carbon and the oxygen. So this all essentially happened because this oxygen had the lone pair. The electrons will fall back, form a double bond. This carbon can only have 4 bonds, so it's going to break one of its bonds, and it's going to be the one right here. So then the lone pairs over here are going to attack this carbon on carbon dioxide. Now, let me just get rid of this. Now we're going to take a look at our final product. And our final product is going to be oxaloacetate. So remember, oxaloacetate, that's where we were trying to get to. We were trying to go from pyruvate to oxaloacetate. Now here we can actually see that, now we can count our carbons. So we have 1, 2, 3, 4 carbons. And oxaloacetate is a 4-carbon molecule. So the whole purpose of this mechanism was going from pyruvate to oxaloacetate. And now you may be wondering, why do we want to go from pyruvate to oxaloacetate? Like I mentioned in the beginning, the purpose of the anaplerotic reactions is to maintain the concentrations of the TCA cycle. So let's say we have not enough oxaloacetate, or we don't have enough of another intermediate in the TCA cycle. we can take pyruvate, convert it into oxaloacetate, so we can regenerate the required concentration of the intermediates needed for the TCA cycle to work. And the process of going from pyruvate to oxaloacetate can also work in reverse. So we can actually go from oxaloacetate to pyruvate. When would that occur? That would occur if we had excess oxaloacetate. The excess oxaloacetate can be removed and converted into pyruvate, and then that pyruvate can be converted into acetyl-CoA, and acetyl-CoA will be fed through the TCA cycle, essentially generating more energy. And that is essentially the mechanism for pyruvate carboxylase. It's one of the enzymes in the anaplerotic reactions from going from pyruvate to oxaloacetate.